Hello and welcome to today's lesson where we're going to be looking at homeostasis principles. By the end of the lesson you should be able to define what is meant by the term homeostasis. You should be able to explain why organisms need to maintain set levels of core temperature and blood pH, and you should be able to evaluate the effect of blood glucose concentration on cells within the body. Okay? So let's start by a word that we have spoken about before, but we're going to redefine it today, the term homeostasis. Homeostasis, okay? Homeostasis is the maintenance of a constant internal environment. So it's the maintenance of a constant internal environment. Now, our environment, or the environment of living things, is constantly changing, okay? But by keeping internal conditions, sorry, I thought I missed a word out then. By keeping internal conditions the same, despite what the external environment is, we can ensure that things like enzymes can work at their optimum rate. So we can say underneath here in red that external conditions, or the external environment, of living things changes all the time. So earlier today I was out in the garden and it was raining initially, and eventually the clouds parted and it went sunny. So things such as humidity, the amount of water, temperature, amount of light would have changed in just that short span of time. But by keeping things the same, so by keeping a constant internal environment, we are able to ensure that the physiology of our body works properly. So we can say that by keeping internal conditions the same, we can ensure physiological processes remain in optimum conditions. Sorry about that, P. Optimum conditions. Okay? Now, physiological, if something is said to be physiological, we mean that it is the internal processes of an organism. So physiological is defined as the internal processes of an organism. So these include things like enzymes or metabolism, okay? So these can include, e.g., metabolic processes, and enzymes. Okay? So we've said that homeostasis is the maintenance of a constant internal environment. The external environment of living things is changing all the time. By keeping conditions internally constant, we can ensure that physiological processes remain in their optimum conditions and therefore happen at the correct rate. Physiological is defined as the internal processes of an organism, such as the metabolic processes and enzymes. Okay? Now, metabolic reactions can be defined as the chemical reactions that take place within living cells. So the chemical reactions that take place within cells. Okay? Now, in order for metabolic reactions to take place, the cells need to have optimum temperature, they need to have optimum pH, and they also need glucose. So we can add underneath here by saying, I'm going to put in regs, it's important, that for metabolic reactions to take place optimally, they need set values of temperature, pH, and glucose, among other things. Okay? So we've said that homeostasis is the maintenance of a constant internal environment. The external environment of living things is changing all of the time, and living things need to ensure that they are keeping all conditions the same internally so that metabolic reactions can take place. Physiological conditions or physiological things are the internal processes of an organism, which include metabolic processes and enzymes. Metabolic reactions are the chemical reactions that take place within cells. For metabolic reactions to take place optimally, so at their best rate, they need to have set values of temperature, pH, and glucose, among other things. So this list is not sort of the be-all and end-all. It's just the three that we're going to speak about today and in the coming topic. However, there are lots of other things that make metabolic reactions either faster or slower. Okay? So this is a video. You can pause it at any point. So let's think about these independently. Okay? So let's think about body temperature first. Something we're going to speak about next lesson in more detail, but body temperature. The optimum body temperature for humans is 37 degrees Celsius. So we can put, and I'm going to use a different colour, you'll see why later, in humans, comma, optimal body temperature is 37 degrees Celsius. Okay? This is so enzymes can work at their optimum rate. So I'm going to put in red that this is so enzymes can work at their optimum rate. Okay? If enzymes are too hot, they denature. So if enzymes get too hot, comma, they denature. At the same time, if enzymes get too cold, they don't denature, but they lose kinetic energy. Therefore, there are less successful collisions, and therefore there are less enzyme substrate complexes made. Do not say that if enzymes are too cold, they denature. They do not. They just slow down, and they slow down to such a point where they no longer carry out the reaction at a rate that we could measure. The reaction goes so slowly we can't really measure it, or it wouldn't be useful for living things. So we can say that if enzymes get too cold, comma, all molecules have less kinetic energy therefore there are less successful collisions with substrates and less enzyme substrate complex is formed. Less enzyme substrate complexes form. Okay? So, in humans, the optimal body temperature is 37 degrees Celsius. This is so enzymes can work at their optimum rate. If enzymes get too hot, they denature, and if enzymes get too cold, all of the molecules involved have less kinetic energy, therefore have less successful collisions with substrates, and less enzyme substrate complexes are formed. Make sure you are using this word enzyme substrate complexes, okay? We are going to speak about body temperature in the next lesson. We're going to speak about body temperature and regulating it in both endotherms and ectotherms. That will be our next few lessons. Today we're just introducing things. Okay. Our next one is blood pH. Now, the blood contains lots of different substances which can both raise and lower its pH. Okay, so we can say that the blood contains lots of substances that raise and lower the pH. Okay? Now, these things are in constant fluctuation. These are constantly changing. So the amount of CO2 changes in the blood on a minute-by-minute basis. The amount of oxygen in the blood changes. The amount of broken down substances, the amount of urea changes in the blood minute by minute, and this can fluctuate. However, your pH of the blood needs to remain constant, so enzymes do not denature. So pH must be kept constant so that enzymes do not denature. Do not denature. Okay? So if pH goes up too high, enzymes will denature. If pH goes down too low, enzymes will also denature. So we can say, I'm going to add in red that any change in a enzyme's surrounding pH from the optimum will cause it to denature. Okay, so all you need to know is that any change in the surrounding pH of an enzyme will cause it to denature. So the final one we need to talk about is maintaining blood glucose levels. Now, we have spoken about this in a previous topic. However, we'll just pick up on the key points that we need for this topic. So blood glucose concentration. So blood glucose concentration should be about 7 millimolar per dm cubed. So blood glucose concentration should be about 7 millimolar per dm cubed. Make sure you put this -3 in the units. That means for every dm cubed of blood that has passed, we have got 7 millimolars of glucose. Okay, it's a unit of measurement, so you need to make sure you've got this minus value at the top. Now, cells need glucose for respiration. Okay, so we're going to put in red that cells need glucose for respiration. Now, it doesn't matter what type of respiration you're carrying out. If it's aerobic, you're going to go through glycolysis and all the following reactions after it, such as the link reaction, the Krebs cycle, and the electron transport chain. Whereas if it is anaerobic respiration, you need glucose if you're in an animal to just carry out the ethanol, not the ethanol pathway, the lactate pathway, and in plants to carry out the ethanol pathway. Okay, but in humans, it should be about 7 millimolar per dm cubed to either carry out the lactate pathway or the aerobic pathway. Now, glucose is transported around the blood and it can fluctuate highly. But before we write that down, before I get ahead of myself, let's get down that if blood glucose drops too low, cells struggle to carry out respiration. So if cells can't carry out respiration, they will start to break down all of the glycogen stores, turn it into glucose in the blood, so that your cells can do respiration without additional glucose in the blood. However, if there is no additional glucose added at that point, eventually your muscles will be broken down, the organs of your body will be broken down, and eventually the organism would starve, okay, and would have no energy for respiration, and its cells would die. However, glucose in the blood can affect water potential, okay? So glucose in the blood can affect water potential causing cells to shrivel and die. Water potential can be defined, just to refresh our memory, water potential, it can be defined as the likelihood of water molecules to diffuse out or into a solution. So the likelihood of water molecules to move out of or into a solution. So if glucose is high inside the blood, it can lower the water potential of the blood. Therefore, water would leave cells by osmosis, causing them to shrivel. So we can say that high amounts of solutes in the blood, like glucose, can reduce or reduces the water potential of the blood. So high amounts of solutes in the blood, like glucose, reduces the water potential of the blood. This causes water to move out of cells by osmosis into the blood, causing the cells to shrivel and die. So we can say that this causes water to leave cells by osmosis and move into the blood. Resulting in cells shriveling and dying. Now, luckily, we have hormones such as insulin and glucagon, which regulate our blood glucose concentration. So the final thing we can say is insulin / glucagon regulate blood glucose concentration. It's the worst blood glucose ever. Blood glucose concentration. Okay, we've spoken about that in a previous topic, so we don't need to go over that in this one. So blood glucose concentration should be about 7 millimolars per dm cubed. Cells need glucose for respiration. If blood glucose drops too low, cells struggle to carry out respiration. Glucose in the blood can affect water potential, causing cells to shrivel and die. Water potential is the likelihood of water molecules to move out of or into a solution. High amounts of solutes in the blood, such as glucose, reduce the water potential of the blood. This causes water to leave the cells by osmosis and move into the blood, resulting in cell shriveling and dying. Luckily, we have insulin and glucagon, which allows us to regulate our blood glucose levels. So if it gets too high, we get the release of insulin. If it gets too low, we get the release of glucagon. Okay? So you should now be able to tell me what is meant by the term homeostasis. You should be able to explain why organisms need to maintain set levels of core temperature and blood pH, and you should be able to evaluate the effects of blood glucose concentration on cells within the body. If you have any questions at all, please contact me by the usual means, and I will get back to you as soon as I can. If not, I will see you in the next video. Thank you very much.